Thank you very much, and thank you to the organisers and the hosts of this meeting for the opportunity to speak today. I'm trying to change topic a little bit now to consider the lens of the eye, and I'm particularly going to talk about some work I've been involved in to do with surveying eye lens doses in the UK medical sector, a small study, and some mechanistic studies on radiation effects on the eye that we've just started. Obviously, a topic of current interest in the light of the fairly recent recommendation from ICRP to reduce the dose limit for the lens of the eye down to 20 millisieverts per year. So my interest in this area really started through review activity. Back in 2009, lead author colleague Liz Ainsbury published with a number of then HPA, sorry, just for clarity on organisations, Public Health England came into being in April of this year, and we were previously the Health Protection Agency. It's the same body, the same people working there. Liz Ainsbury and colleagues did a review looking at primarily the accumulated epidemiological evidence of sensitivity of the lens of the eye, and it concluded, as you can see there, that the threshold dose was clearly less than the previously set 2 Gray level. It was more in the order of half a Gray, consistent with the tissue reactions report. And it may, note may, not is, be more accurately described by a linear non-threshold model rather than a threshold-type model. But there's great uncertainty on the mechanisms that apply to radiation cataractogenesis. More recently, following the publication of the ICRP statement, we published a review to update to the review to include some further epidemiological analysis, which still suggested a threshold dose of half a Gray. There was no change to that. And we believe there's strengthening evidence that stochastic processes may contribute to the development of cataract following radiation exposure. But in the end, we came to the conclusion that the ICRP made a sound judgment in recommending a 20 millisievert per year occupational dose limit for the eye lens. So this really raises two major issues, and the recommendation of a lower dose limit did raise some concerns, particularly in the medical sector, that this might present some problems to implementation. So the two issues I want to talk about are mechanistic studies and a small study of radiation doses to the lens of the eye in the UK medical sector. So first, the dose survey. So we looked at 3 UK hospital radiology departments based in London, Oxford and Manchester, supporting a full range of interventional procedures, interventional procedure cardiology as well, over a 4-week period in January of this year. We used a eye lens dosimeter, which is shown in the picture here, provided by PhD colleagues in our dosimetry department, and it's supported by a questionnaire which took into consideration the job role, the procedures carried out, and the PPE, personal protective equipment, used in the practice. And this was a study funded by the UK Health and Safety Executive. So the results of this study, 61 dosimeters were returned, so we had quite a good uptake of those we sent out. The median dose of those dosimeters was below the detection limit of 0.15 millisieverts for the period. Only 13 of the dosimeters returned had levels above that 0.15 millisievert level. There was no correlation by the type of procedure, the number of procedures, or the PPE use, and the dose actually recorded. So there's clearly heterogeneity in practice and heterogeneity in the eye lens doses that individuals receive. And generally, most practitioners appear to be protecting themselves quite well from the eye exposure. However, there was a maximum dose recorded in this small survey. It was 0.16 millisieverts in the 4-week period, which would, if you extrapolate in the simple fashion, give a dose just slightly over the 20 millisieverts per year. So there is some issues perhaps to address. In terms of analysing the questionnaire, the survey included a mixture of consultants and registrars, particularly focusing on those groups as those who were predicted to have the highest exposures within practice. And there was a total of over 1,000 procedures looked at of 20 different types. So it's quite a wide-ranging survey of the type of procedure looked at. The median number of procedures that each practitioner carried out was 15, but the range was really rather wide, between 1 and 70. Generally, the uptake of PPE use was very good, but there was rather notable the sort of fairly low uptake of the use of lead glasses. Only 9 of the participants reported the use of lead glasses in their practice. The dosimeters were worn as per instruction, so that's on a headband above the eye. And another aspect I'm not really going to talk about much, but dose area product information for procedures was gathered, and that is potentially useful in monitoring doses in the future, we believe. So the conclusion of this study, which is limited because it's not very large, it's only 3 centres in the UK, and obviously there's many, many hospitals in the UK, let alone worldwide, give a generally reassuring picture. The targeted high-dose procedures in 3 hospitals, over 1,000 procedures screened. Doses received varied depending on a number of different factors, and there was only 13 giving doses of potential, registrable doses, if you like. But generally, practitioners didn't use lead glasses, and if we extrapolate from the highest doses we've recorded, there was some potential for a limited number of people receiving doses above the 20 millisievert limit, but that assumes an equal workload across the year. This study is now published, accepted for publication in the Journal of Radiological Protection. There's a lot more detail on the procedures covered and so forth, which you'd be able to read in the near future. I'd like to turn briefly to the mechanistic studies we've carried out to supplement that dose survey information. So here we used primarily an animal model, the mouse model, C57 black mice exposed in vivo, supplemented by cell culture studies of human and mouse lens epithelial cells in explant or in cell culture, exposed to a wide dose range, 20 milligray to 2 gray, looking at early and late events, changes in those systems, looking at multiple endpoints as well. And I think the particular point I'd like to emphasize, looking at early and late is very important in these mechanistic studies to extrapolate from what you can see very early to what occurs during disease development. Just to give you a flavor of some of the preliminary results coming out of this study, looking at DNA damage through gamma H2AX assay, which is a readout of primarily DNA double-strand breaks. These are sections through lens epithelia from the mouse, sectioned, and the green staining shows the presence of DNA damage. And while there are some technical problems with this assay still, we can see dose responsiveness down to levels below, certainly below the threshold doses that are now recognized. Looking at a different endpoint, effects on cell proliferation. Cell proliferation is quite important in the maintenance of the eye lens structure, clearly. Looking at early time points after irradiation, there does appear to be some potential discontinuities in dose response. There's a dose response series across the panel from left to right, again, looking at 24 hours and 1 month after exposure. The important point I wish to make here is that the response one sees at 24 hours, which may show some discontinuity in dose response, is not necessarily seen at later time points as well, clearly emphasizing the need to look across a wide range of time points in these sort of studies. So, these are preliminary results, and there's a lot more work going on in this area. We'll be looking additionally at morphological changes to the eye lens during fibrogenesis, looking at cell cycle arrest and cellular senescence, 2 other endpoints that have been suggested to be important in the development of cataract. Also looking in other tissues, we're looking at the radiosensitivity across those different tissue types. So in this very brief presentation, I hope I've shown you that there is the possibility of doing eye lens dose surveys in hospital settings in a very effective fashion, and there are routes into trying to understand mechanisms a little bit better on the basis of animal and cellular models. Finally, I'd just like to acknowledge my colleagues who have been involved in these studies, Liz Ainsbury, Jackie Gillen and Margaret Costa from my own department in PHE, Phil Gilvin, who runs our dosimetry service, Sarah Peters and Kathy Slack, who work in our medical exposure department. Very important to get buy-in from the hospitals in those dose surveys, and those colleagues were very important in getting those dosimeters out and effectively returned from all participants. The mechanistic studies were carried out in collaboration with colleagues at University of Durham, University Eva and Roy Quinlan. And as noted there, the funding from the Health and Safety Executive for the dose survey and from the Department of Health Radiation Protection Research Programme for the mechanistic